Hello, I'm Per Ahlström, calling from Sweden, from Uppsala University. I'm really sorry I can't be with you in person today, but I'm grateful to the organizers for giving me the opportunity to give a talk. And I'll start by sharing my screen. So I'll be talking about taxonomy as the foundation of biological research. And traditionally, taxonomy has almost exclusively been carried out in museum collections, where researchers have lined up large numbers of specimens and tried to group them based on their appearances. And then they have classified birds that have similar appearances as the same species, and birds that look different, they've treated as different species. This is, of course, a grave oversimplification, but basically you could say that that's how it has been done in the past. So museum collections are still of paramount importance for taxonomy, but there are also lots of other tools available to us today that were not available, say, 100 years ago or even 50, 60 years ago. So I'm going to present to you the modern approach, which is often called integrative taxonomy. And this approach, it tries to integrate multiple independent lines of evidence of lineage separation for which, well, under the phylogenetic species concept or the broader, more inclusive general lineage concept, or of reproductive isolation, which, as you all know, is the hallmark of the biological species concept. So you can use lots of different types of data here. You can use morphology, both plumages and structure. You can use DNA, single genes, or even whole genomes, which are often available today. Vocalizations, really important. Songs are generally considered of higher importance than calls, but both types can be used. Other behaviors, in particular, such behaviors that are of importance for mate choice, such as sexual displays. Ecology, including habitat. Geographical distributions, including different relationships such as sympatry, parapatry, allopatry. And species are considered hypotheses, and therefore they are testable. And you can test them by using all these different lines of evidence, as much evidence as you can get. So the more data you have, the better. The more different independent lines of evidence you have, the stronger your case will be, the stronger your taxonomic decisions will be. And I would like to stress that this really is the recommended way to work, no matter which species concept you may favor. So I will present a case study, namely the Zoothera mollissima species complex, the Plain-backed Thrush complex. And if you want more details, you can look at this paper, which we published a few years ago. It's an open access journal, so you can quite easily find that. And when you embark on a taxonomic study, like with any scientific research, you need to collect data. And it's crucial that you study samples from throughout the range of the species complex that you're studying. So as you can see here on these two maps, which show our sampling for the plain-backed thrush complex, we've sampled really throughout the entire range of this complex. And we've collected lots of data in the field. So we studied plumages, structure, bare part coloration, behaviors, etc. And we also took DNA samples in the form of blood samples from the birds that we caught. And in the picture on the left there, you can see one of the important co-workers of this study, Shashank Dalvi, who some of you may know. We also studied ecology. So we looked at habitat differences, and we could see that different populations, which seemed to differ in appearance and very strikingly in vocalizations, they occurred at different habitats. So, for example, one population was found in the alpine zone above the tree limit, really, really high up to more than 4,200 meters. And other populations were found in different forest habitats. So it could be both mixed forest and pure deciduous forest. So there's a very great elevational span here. And these populations we found were actually segregated very much by habitat and elevation. And in some places, I should say also, we found different types occurring in the same area, but segregated by habitat and elevation, both in China and in India. We also collected lots of sound recordings, and we produced sonograms of these to look at fine details and to analyze different parameters. So sonograms are excellent for finding differences among different populations, and you can easily take measurements of different time and frequency parameters, for example. And we did that, and we could also hear that the songs were quite different between different populations. I'm going to play two of those populations for you. The first one I play is represented here in the sonograms by the top 6 panels on the left page here. And it sounds like this. So it's a very scratchy, unmusical song, and you might wonder, how can a female be attracted to that? The other one, though, I'm going to play is from the forest area, so below the alpine zone, and it's here represented by the 2 sonograms at the bottom left and the 6 panels on the top right, and it sounds like this. So a much more musical, pleasant sounding song. And we also used the measurements that we took to quantify the differences between those. And here is a plot based on a multivariate analysis, where you can see that different song types cluster very tightly, but they differ clearly from each other. And we also, of course, visited museum collections to look at specimens there. And in particular, we wanted to see all the different type specimens that were available. So these name-bearing types are, they represent the name, so the link, the name that's been given to population to a specimen, so you know what the name actually refers to. And therefore, it's really, really crucial when you're doing taxonomic work to look at name-bearing types. So one of our co-workers, Pam Rasmussen, she probably looked at every single specimen in the world, almost, of these thrushes in different museums. And she did really meticulous, careful analysis of morphometrics and of plumage characters. And as you may know, Pam is an excellent person for this type of job. Another excellent person is Urban Olson, who gave a talk yesterday, I believe. And he sequenced not only the DNA samples that we collected in the field, but he also sequenced museum specimens. So you can actually get DNA out of museum specimens, and we got little samples from the toe pads of the birds. And we tried to include all the different name-bearing types that we could find in different museums. Those are the ones that are highlighted in bold in the text here on the graph. And as you can see here, the plain-backed thrush is divided into 4 different deeply diverged clades. They are represented by the different colored, the different colors here. So these clades have been separated for millions of years. You've got a time scale at the bottom there, and they've been separated for a really long time. And you can also compare with 3 other species at the bottom there, which are clearly different from the plain-backed thrush and from each other, and they've always been treated as different species. So if you look at the time comparisons there, you can see that these plain-backed thrushes, which had previously been considered a single species, are really very, very different genetically. So our conclusion here is that we identified 3 main groups, which we considered to be very clearly 3 different species. And we found corroborating evidence from morphology, song, DNA, habitat, and we also found symmetry between pairs of those in different places. And we identified which names should be used for these. So one of these groups is the original plain-backed thrush, so the Zoothera mollissima sensu stricto. And we propose that this should be called alpine thrush because it's the one that occurs at really high elevation in the alpine zone. That's where it breeds. In winter, it comes down to lower elevation. And then the bottom one marked in yellow there is the Sichuan thrush, which we used the name Zoothera grisaceps for because that name was available. And then we realized that one of those did not have a name. So we had to name this Zoothera salim alii in honor of the famous Indian ornithologist Salim Ali. And that's the Himalayan thrush, which occurs in forest mid-elevations. And then, as you can see on this phylogenetic tree, we identified one more group, which we tentatively called Yunnan thrush. This is only based on DNA sequences from museum specimens. So we think that we found it in the field, and we think that it's got at least a slightly different song from the alpine thrush, but we need more information before we can come to any conclusion regarding what that is. So what was previously considered a single species, the plain-backed thrush, is now treated as 3 different species, and it could even be 4 different species. And one of them named in honor of Dr. Salim Ali. So I'd just like to touch very briefly on computer-based species delimitation based on DNA, which has gained some popularity in recent years. So you could ask, is this all you need? Is this a magic wand? Is there any need for integrative taxonomy? And I would say, yes, we do still need integrative taxonomy. Computer-based analysis, computer-based species delimitation can be used as part of an integrative taxonomic approach. But you can't just trust what the computer tells you. So if the computer tells you that you've got 3 different species here, you still need to evaluate that based on independent data. So morphological, vocal, behavioral, and other differences are actually good additional evidence of lineage divergence. And they are actually faster evolving than most genetic markers. So they may show evidence of divergence long before most of the genes show any evidence at all. And also the genes coding, for example, some slight morphological traits, they make up just a tiny, tiny fraction of the genome. So they become totally swamped if you sequence a large part of the genome. And also sexual signals, which are important for reproductive isolation. They can evolve rather fast, long before they become evident in the genomes of these birds. And also, it's quite possible if you're using a computer-based modern system that there could be 1 or 2 loci out of the number that you're using that might have a really strong signal compared to all the others, and the others might be conflicting with these 2, but not so strongly, and they might be conflicting among each other. But these 2 really strongly, these really strong loci might still provide an inaccurate phylogeny, for example. So I've tried to convince you here that taxonomy is really, really important, and it's crucial for studies of different aspects of evolution and distribution, ecology, I would say almost every aspect of biology. And if you have an improper knowledge of taxonomy, you might actually come to the wrong conclusions regarding lots of these issues. And integrative taxonomy, that's the way to go in these cases when you're doing taxonomic work. So thanks very much to the co-authors of this study, and to the ones who provided photographs, and to you for listening. And I will be available to answer some questions. And if you want to get in touch, you've got my email there, and there are also 2 links to web pages where you might find some additional information. Thanks very much.